Now here's a new method. In fact, it was the one that was developed by Tanya Goldberg. Why we want to predict is because sub-cellular localization tells you something about function. And in fact, here's another aspect. So ribosomes is not really a sub-cellular localization, it's a machine. A local molecular machine, and there are some of these machines that in fact also are functionally important. So being part of the machine is important because the machine does express proteins, and because what you do as an enzyme may depend on whether you're part of that machinery or some other machinery. Okay, but let's recap log3.1, and I made a mistake here. Log3.1 was the system of SVMs that is somehow loosely based upon the hierarchy of the molecular sorting pathway. And now the question is, how can we improve over that? So what's the signal? And I told you that one of the the advantages of this type of tool is that by introducing this particular case with SVMs, different SVMs for different decisions of that process, for instance, the first one being this one is secreted or not secreted, we can look at different input signals. That's the strength of the system. The system as it was like this had a number of states. That number of the number of states shown essentially here. In fact, it left out, for instance, membrane proteins. So one idea of going into the next round of log3.2 was to bring membrane proteins back into the game. But what else could you imagine to improve in terms of the method of what I have presented to you so far? Any idea? So one important point that I've not presented to you, and I will also not present with log3.2 and 3, is that we should find a way to combine everything that we have. I talked about different ways of solving the problem. I did not tell you how to integrate all of these. And there is, at this point, still no good way of doing that, still no good solution, so that remains an open problem. We have seen in the development of log3 that bringing the alignment information back in was very important, but it's done in a hack sort of way. And the typical hack sort of way is PsyBlast gives a clear result, we take it. PsyBlast doesn't, we take log3.2. That's the way we do it at the moment. Again, now in terms of method here, the idea was to bring in some other information. So, but first really to bring in more states for eukaryotes. And that led to the decision of, in fact, the first step going between membrane, non-membrane and membrane. So, as opposed to going secretory pathway, not secretory pathway. And you can clearly understand that there is some rationale for that. Now let's just go over these introductory slides. On which I may have spent a little bit too much time. So the method idea. That's the publication, and the publication talked, those are different ways. I again said we didn't combine them. So log3.2. The idea is to have one framework for all types of life. In some sense, what I showed you before is a framework. You have a hierarchy, you use SVMs, and you can consider that the framework. Here we go another step by simplifying the process of using the input features. I'll get back to that in a minute. Here is an aspect that in fact we try to address explicitly. That's robustness with respect to sequencing errors. You may remember that one of the aspects of sub-cellular localization prediction was always we use the entire amino acid composition. As you can imagine, if I start now with a stretch, either because I have the wrong domain or because I have the wrong reading frame, so I have actually a fraction of the protein, this is likely to go very wrong. So how could you imagine to in fact solve this problem and become more robust sequencing errors? Any idea? So again, the problem is that sometimes sequencing errors lead to mistakes, sometimes they lead to missing entire introns and sometimes entire domains. In fact, sometimes the type of method that we have is queried with one domain. It's a PDB protein. It's a subset. The protein may have 10 domains and one is taken out and searched through the server, and the server is not really optimized for that. How could you imagine to have a server that sort of addresses that issue directly? No idea? So the problem is that the thing is too long, right? We shorten it. We look at the locomotive. We cannot do sliding windows. So some of you, you were the last, no, you were not there last year. So one of you was in the room last year, and for most of the efforts that we did in order to predict three-dimensional structure, two-dimensional structure, 1D structures, so secondary structure and the like, we use windows of, in the ballpark of 15, 13, something like that, consecutive residues that we move through a protein. And every single one we ask, is that central residue in a helix? That's typically the kind of design that we don't have here. We could imagine that we, by sliding windows, could find motifs, nuclear localization motifs. In fact, we tried, we failed. But we need something somehow in between these two worlds. Now, we want in fact to have 18 different classes for eukaryotes, 6 for bacteria and 3 for archaea. We want to have membrane predictions built in. We want to use this idea of a decision tree because it worked. We want to keep reliability, and we want a high performance. So data set preparation, we have, in fact, those were the numbers I said before. We have 59 sequence-unique proteins for archaea, about 500 for bacteria, and about 1,600 or 1,700 for eukaryotes. So this is not that big a set. So we have to be careful with the way we, that is, we take the data from SwiSprot, the way we in fact train our system. That's the background for the homology-based assignment, and for the homology-based assignment, we know that we have to reduce the data set. We know stratified k-fold cross-validation. So let's get one step back. K-fold, in this particular case shown 5-fold. You have the large set, you cut out some cross-training on testing sets. So this is just the old-fashioned two types of data set, which today for most things doesn't work, but let's just assume for simplicity that would work. In that case, we have a gray training set and the cut out cake here being the test set, and then we rotate around such that every single set has been used for testing exactly once. Now, the part that we discussed about, the aspect that we discussed about many times, is that between testing set and training set there should not be an overlap, in the sense that I could in fact use one to predict the sub-cellular localization by homology-based inference to the other. That we have been clear about. Do you know what the word stratified adds to all of that? Have you heard of stratified cross-validation? So the idea now is, in fact, that the composition, the reality, input reality in the gray reflects the input reality in the dark gray or black. Essentially, that you create a background such, you do a cross-validation such, that in fact these two data sets essentially have a similar background. Why would you want to do that? Why is it good? And what is the problem with it?
Stratified for the different cellular compartments, where the proteins are localized?
That is one way of looking at it, yes.
Okay, so I would say that...
In fact, that's, I don't know whether that was the point that, oh no, it was not written on this slide. Okay, it was written on the original slide from Tanya. Yes, it is written here.
So you want all your training sets to be representative for the entire set. So you don't want one training set to be made up of proteins which are only localized in one specific compartment.
Mm-hmm.
So,
well, you look at the extreme example. So let's just look at an example where in one split, here I would have 50% of nuclear and another one 55. So whatever you say, this is not the idea of stratified, because stratified would say they have different compositions, hence they don't partake in this idea of stratified. But in principle, they would still not violate what you say. So for that, for what you said so far, it doesn't really have to be enforced to be identical, or close to identical, right?
At least you have to know how the composition in the training set is in order to assess your prediction.
Correct, that's correct. But again, the idea of stratified is that you have equally sized. So why would you want to do that? The point is that it could happen that if you didn't do that, and I'll show you later in the lecture an example for that, that the system, the machine learning device, may zoom into some aspect that is a happenstance aspect. So by having those two distributions equal, you reduce the probability that the system will learn this as special for the future. Okay? That's the upside, the upshot. The downshot is it will implicitly learn some new thing that is special. And the new thing that is special is the equal distribution, which in fact is possibly not at all reality. And that may lead to completely misleading classifiers. You have to be very careful about stratified cross-validation. Okay? There's a good reason for doing this, and there's a danger in this. Yeah?
But you could construct it that way that the distribution of each stratum resembles that of the entire population.
Yes. Now we get into the ultimate problem. The ultimate problem being that you have always a distribution today. You do not quite know to which extent the distribution today is in fact relevant for the future. It may very well be true that the way we classify nuclear proteins is different from the classification of secreted proteins and depends on the type of annotation tools we have available today. That in fact, for instance, just to make the extreme point, I don't see the proteins in the body, in the blood. What I do see is only the classification or the sub-cellular localization compartments inside of one cell, because I grab out a cell. By this method then, I take out a cell, grind it up into its components, I will always miss some secreted proteins, because they're not part of the cell, right? They're out already. And simply by that experimental bias, what I have today may not be the thing that I will have once I, in fact, do this whole thing to an entire body, right?
Ah, excuse me, it's already written there. Yeah.
That is something that you have to be aware of, and you cannot, in fact, do that by just making sure that these that these different splits, in fact, have identical compositions, problem solved, and that if they all have identical compositions, then all have the same as the entire set, but that may not be the truth. Okay, so take switchpot, we do training testing, and in fact, here's an additional issue illustrated. What we have to make sure is that between testing and training, there is no significant sequence similarity, right? So there should not be a line here between the blue and the points up here. However, in many cases, you can bring in some similarity here. Say the clustering at whatever your threshold chosen was, h-val 0, h is an e-value of 10⁻³ gave you these blue points. And then you simply group these blue points, and 2 go into testing set, and 6 go into training set. And say you find out that 6 is just not enough. So what you then can do is pull some red points back in that are such that you could, by homology modeling, if you had that, predict the sub-cellular localization of this one. But this one may still add an important sequence signal. You can do that as long as none of the red ones you add overlaps with the blue in the testing set. Right? That's a mechanism that is very often used, and in this particular problem, we use that. Is that point clear that I made? So again, let's get back. There are two reasons why I need to redundancy reduce those data before I split into testing and training. And again, I need 3 data sets, cross-training, training, testing, but let's just explain it in a simplified view. Reason number 1, I have to make sure that nothing in the testing set is sequenced similar to anything in the training set beyond the point where I could just use PsyBlast to do the prediction. That's statement number 1. Statement number 2, I have to bring into my machine learning device a representative distribution. A representative distribution means I should not take the experimental bias that we know exists in any data set I create. That is sequence redundancy reduction. They are related, these two things, but strictly speaking, they have different rationale, a different objective. In terms of the way of doing it, at the end of the day, they are similar. So you typically take your data set here, cluster, and here in this particular cluster I have 2 different types of clusters. One is the HVL0 cluster, is blue. One is the HVL60 cluster, it's red. Looking at the blue, that I herewith postulate is the cluster that I need to do in order to have a clean separation between testing and training. I can use any grouping of the blue ones in testing and training at the same time because they're far apart from each other. So whether I do the green here or do the green here, doesn't matter, right? According to that distribution. Now, I have the problem that I don't have enough data points and I cannot do new experiments. Is there any way to go through these data again, giving me the hope that I get a little bit extra? And the answer to that is, let's go back to the 60, the HVL60 clustering, the red ones. The red ones are such that I cannot have a blue and a red here in training and testing. I cannot have the green test set here and that blue training set because they're too similar. But I can have, and I can also not test on red ones because that would overrepresent a certain part of the distribution, which is the second reason for clustering. It may be that I test too much of nuclear proteins or too much of one particular nuclear family. So I can only test on blue ones, to be clean. Now I need more for the training, so I can pull red ones into the training set. They bring redundancy, but it might be that this redundancy is actually helping me. How would I know whether bringing these red ones back in will help? Again, I can only do that as long as I make sure that none of the red ones here is connected to a blue one in terms of HVL0. So how do I know whether that's the case? Whether I benefit? It's very simple, you try out. Okay? There's no other way to know. You see whether pulling in at some level of HVAL, it helps you. And for the method that I talked about before, and much more importantly for this method here, it does help you when you go to a level around 60 or 70. Much beyond that, it doesn't help. Okay, now let's get back to the issue of training. And linear support vector machine essentially introduces lines that classifies, in this particular case here, red and blue. It does that by finding a margin that is such that within that margin we have the most similar blue and red, most similar to each other. So the way the SVM can handle very little data or a few data points is by zooming into the closest ones and by making the decision how to separate them such that it optimally separates out the ones that are very close to each other. And what do you do if separation is not, if the problem in this particular case here is not separatable, we essentially project it into a higher dimensional space in which the problem becomes a linearly separatable problem. That's done through the kernel function. And there are different types of kernels. There are string, subsequence kernels, mismatch kernels, profile kernels, and they're used for different problems. In our context, we used here the profile kernel. The profile kernel goes back to Christine Leslie and Bill Noble, a 10 years old publication. Let me just illustrate this idea. So you have two proteins here shown, the sequences. You look at, what we're looking at is k-mers of 3. And let's assume we look at a threshold of conservation of 5. So what we're going to do is we're simply going to look at all 3 mers. All 3 mers, we build up a vector that describes the first protein that is built up of all possible 3 mers. Okay? And for these particular 3 mers, we simply ask how often is in that 3-mer in my alignment do I see conservation by some criteria. The threshold 5, in this particular case, in this particular example, will lead to a situation in which the CAW is excluded, the core, and the exclusion of the core. So essentially we have 3 1s here, all other zeros. I do the same thing for my other protein, and then look at the vector product of these 2 vectors. That gives me the value. That's a classification, a profile kernel classification of the problem. And then now all I need to do is essentially apply this. The way I, in fact, by the way, going back to what's the best k-mer, what's the conservation threshold, those kind of things I answer by testing with some subset of the data. I get something that is in a reasonable ballpark. And then I use that for the classification of my data points. I have a kernel selection of the parameter combinations. And in fact, when I look at different types of kernels, we see that the profile kernel is an overall percentage accuracy for different classes. You may observe this one we did for eukaryotes in 10 classes. So you try to make all of these decisions. What type of kernel, what type of device, how to set 3 versus 5 versus a k-mer and a threshold. All these kinds of questions you try to answer with a problem that is not exactly yours. You use a subset of the data, and in this particular case, remember, we are going to use 18 classes. Here we just use 10. Again, all of that goes in a direction where you try to avoid to go into a space where you have an optimization that is valid only for your problem. You try everything to have something that is relevant for your problem. We're talking about sub-cellular localization, but a very different mapping of sub-cellular localization. This may not give you the highest performance for your application, but this is likely to give you something that will survive longer. So more conservative, okay? There is still here is an average of a training set done. I would argue not to do that, because we are still at this point in the process of making the point, let's find a new method. Let's not just take the regular SVM that we took so far and play around with the input. Let's build it on some new feature. So in this particular case, the answer now is we're going to use the profile kernel. The next issue that we have pertains to the question of how do we... Hi?
Sorry.
It's open. Wasn't it? Did you try? Did you try to push?
Nothing. Yeah, I tried, but I couldn't.
Oh. Okay. Oh yeah. So Jonas closed it. It was open before Jonas came. No, that cannot be true. He said it doesn't. Anyway, so again, the problem that we have is, by the way, another important difference between neural networks and SVMs, although the math behind it is very similar, is that SVMs work essentially on binary classification. How can we bring the problem of binary classification into the world of 18 different classes? And the answer to that question essentially is referred to as multi-class classifier. The first one here really is one against all. The way you do that is essentially the same thing that I showed you before goes back to Wapnick's first publication of SVMs. You do one that says this is nuclear versus everything else, and so forth. Here you can decompose your classes into a system of nested dichotomies. I'm sorry. And every nested dichotomy is equally probable. And then that means that in fact at the end of the day you're left with the option which one to take. And there your answer typically is you take an ensemble, you take several. You combine whatever you have, hoping that this, again, you reduce random events that happen through the training procedure, right? Another way of doing that is you look at an ensemble of class-balanced dichotomies or of data-balanced. So class-balanced is again what we had before. The idea of stratified, so we have in fact every single of these 18 classes done equally, and you immediately see for 18 different classes in eukaryotes this is going to be difficult because 18 classes will absolutely not be spread equally. You could have a data balanced way of doing that in which you have in fact different nodes in the tree that have a different way of balancing it. And then there is the nested dichotomy with a particular fixed architecture that essentially, and by that one you already see that we are going in that direction, that essentially tries to mimic, in this particular case, the biological shuttling of proteins through the cell. Now, the issue that immediately is different from the cell transporting mechanism is we decide on membrane versus not membrane up front, which again, you can actually also argue that coming off the ribosomes, you in fact do very often go into membrane-bound compartments transported. In more detail, essentially then, every one of these decisions here is based on a profile kernel-based SVM. So the first one does membrane, not membrane. This one goes then next into the secretory pathway. ER is the first one, Golgi versus rest, and the PM here. Here we go first, the first decision is nuclear. That in fact is, again, because it's gated, but it's also because that is the best decision that we can make at this point. For all the non-secreted proteins, the distinction between nuclear and rest is the one that, because of a variety of reasons, ultimately because of the NLS, works best, for which we have the best classifiers. That is here the reason we can put the biology on top of it and say that is a gated transport, so it stands out. But there is no, for this hierarchy, there is no direct biology anymore. So that's why this went on here. The chloroplast in the end, mitochondria, that is a decision that is very plant specific. It's another thing whenever you develop a method and somebody sends in their sequences sitting on the web and you predict it's chloroplast and that is a human protein, be careful with these kind of things. Be as far as you can. This is the kind of thing that can seriously hurt your reputation. In fact, one of our systems had a discussion in a high top tier journal that was bashed because that was the prediction at the end of the day, that for human proteins, in fact, it predicted a lot of chloroplast. There was a reason for that, but we should have written at least some flag in the output. We know this is human. We know human doesn't have chloroplast, but we argue there's a reason for that. This is, when you develop methods, think about things like that may be obvious. Okay, now again for the prokaryotes we have a much simpler system. We have a cytosol, extracellular space, and the membrane, and essentially, so shown here, we have the three different compartments, and that's as far as I can. So sorry for my emotional way of looking at the slides. I'm working on this since 6 o'clock in the morning, and all I had was a PowerPoint slide, and all I did is transfer PowerPoint to what I felt is slightly nicer, non-PowerPoint version. That's where the entire time went. And I'm wondering whether it was worth it. Now we're back to the original PowerPoint talk from Tatjana. In bacteria, so archaea have 3 different classes. Bacteria have on top an outer membrane and have a periplasm, so have different classes added to them, and that is the view. On the eukaryotes, again, 18 different classes. The first decision is here. I went already through these. Sorry. Now, I went through this. The question now becomes, how good is the system working? In terms of answering the performance, there's a Q18 number, so 18 states, accuracy 65%. Okay. Is that impressive, is it not? Do you remember any of the numbers that we presented so far? Ballpark figures? Have you seen numbers like that before? Do you have any intuition? If I had a four-state system that says cytoplasm or cytosolic, secreted, mitochondria and nuclear. If I told you I have a method that is 95% accurate, would you believe me? On 4 classes. 4 classes in eukaryotes. So I take it. Yeah?
Well, the expected value, if you just randomly chose one label, would be 35%, right?
No, wrong. But would be the lower bound of the answer would be 25%. That's true. Since the classes are not equally distributed, the number is higher, substantially higher, actually over 30. Yeah. Okay.
So
it's much better than random.
65 is much better than guessing.
That is a very, very, very important statement to make. So whenever somebody asks you a question in an environment where you have essentially nothing to say, try to figure out what you can say. And that is the first statement that's very important. 1/18 is a number that is much smaller than 65. That's essentially what you say. And that's whenever you see some number in the newspaper about some wars or whatever it is, try to do some simple thing and reflect on numbers in this way. That is scientific procedure. You're totally right. So 65 is way better than random. So do you have a sense for, let's get back to the Q4 is 95%. I claim that my method does that. Do you believe me?
I think it's too good to be true.
Yes, because 95 is too high? What about, okay, so it's not true. So that no method gets to that point. But if I said 88, for those of you who have been around last semester, you were, right? No, oh, you're getting more and more of a minority. So 88 is what you get if you predict secondary structure through homologous proteins. So if you have 2 proteins that essentially have the same 3-dimensional structure, then they agree in the 3-state secondary structure, helix, strand, other, by about that number, to about that number. That is sort of 88%. So, is that? So, quick answer is, at a psi-blast E-value of 10 minus 10 or something like that, you do not quite get that. So it's the number is still substantial, still a little bit lower than that, but that is the ballpark. For some proteins we can reach that through PsyBlast we can reach that ballpark. PsyBlast for these, in fact, does only better for a few for which it works. The numbers I should present to you, I don't. But this is much, much, much higher than the methods that I showed you before on something like that. It's an outstandingly high number. In fact, it was so high that in fact, when I saw that number, I didn't believe it. And again, to some extent, based on our experiences with sub-cellular localization, some extent based on 100 by 18 and being so far away from random. Now, here, since we have this hierarchy, dichotomy, we in fact can also ask how are these individual decisions differing? And the first question here is this one. I distinguish between transmembrane and non-transmembrane proteins at a level of 94%. Is that good? I understand that we have a minority issue here now, but for everybody in the room who has heard about transmembrane prediction, is that something that we can achieve with transmembrane? So just a simple feature such as we have a segment or a long segment of hydrophobic residues. We call that a membrane helix. If we have it, we call it a membrane protein. Does that kind of simple thing reach this level of accuracy, you believe? Any idea where that would go? So what I just described to you as an algorithm, just using hydrophobicity, an ongoing run of hydrophobic residues, and say whenever I have that, I call it the membrane proteins, does lead to 92% about, but not 92% accuracy. There's 92% error. So that leads to about 8%. But intelligent membrane prediction methods, they reach levels that use also combine Spocticus is one of those, and now I'm blanking on another one that in fact is the best current method by our view, that uses signal peptides and membrane helices together in a prediction system, and they reach levels of prediction accuracy just below this. Repeat, this is a fairly high number. If even the best polyphobia, it reaches just this number.
Is that phobias? Phobias.
Polyphobias. I just put it down and I made a mistake. I thought it was below, but it's actually not. It's actually higher. I made an important mistake. So polyphobias, in this particular context, gives a higher number. I'm not entirely sure whether that is the number for the same data set. Because the number that I believe, I have to look it up. Anyway, so this is a fairly good classification for a system that has no idea about membrane helices. And then you go further down in your classification tree, and as was true for the last system, the further you go down, the more the numbers go down, because simply you, in fact, make more difficult decisions. It's also true that at this point there's a technical issue for going further down. There is also a technical issue. I don't know which of the two dominates, whether it's the biology or the technical issue, which is the further you go down, the less data you have, and the more the data is polluted. You could call that a technical issue, you could call that has to do with biology, that things are more on an exchange the further you go down, less differentiated. Or you could just say, right? Yeah, thank you. Thanks. Or you could just say that it's just a database issue, and to some extent only time will tell which of the two are true. This graph from Tanya looks at the view from different angles, so accuracy, how many of those that you classify in the nuclear against the rest of the data, rest of the world, right, and coverage, how many of the ones that you could have found did you find at that level? And those are, in fact, coverage is in green here, this number, and this is in terms of accuracy. You always need to somehow show these two numbers. That, by the way, brings me to another issue that I have discussed and I somehow glossed over on my other slides, I don't know why, which is the question of how do you estimate errors? Yes?
Yeah, we do cross-validation, right? So we have several runs.
Yeah.
So we get
Everybody happy with that answer? That is one way of doing it. You're right. It's not only one way of doing it. That's in fact exactly the way that packages out there would apply. And it clearly is an easy way, and it's a cheap way in the sense that you don't have to do anything else. So you simply look at the difference between different cross-validation splits. They give different levels of performance, and the difference is directly one measure for the degree to which the data is not consistent, to which you in fact make an error in the estimate. That's absolutely true. Any comment on that? Well, assume that we have a very large data set, and assume that we don't do that many cross-validation experiments. So we just make 3 because of time, because of CPU, or whatever the reason is. Then we have a difference between 3 different data sets. Each of these data sets is based upon a large data set. Okay? That leads to a situation in which your estimates for error are on the lower end of things. You essentially want to ask or answer the question, how much would it change if I remove 1 protein or 2 proteins or a few? Not if I remove 500. The larger your data set, the smaller the number of k-fold, so the larger each split, the more likely these splits will not differ. Hence you will actually estimate an error that is fairly low, because you will not see a difference. Simply because your data sets are so large, right? So the cross-validation lookup works as long as your data is really far away from saturation. Or as long as you do a k-fold cross-validation where k is a very, very high number. So rather than what I showed you 5-fold or 10-fold, on some of these slides you may have seen that, if you did for 1,000 protein 1,000-fold, so they leave 1 out. Then again, this procedure is safe, but typically that is not safe in terms of CPU. So, can you think about another method of doing it? Yes?
I'm thinking of bootstrapping.
Yes. Anybody? So he is thinking about bootstrapping. Can anybody else divine what he's thinking about? What is bootstrapping? Do you know where the so that is on one of my slides that I glossed over for the sake of just running through slides. Actually, Tanya's slides, and at least. Oh no! I didn't realize this. That cannot be that far away. Okay, it's somewhere else. Okay, so yeah. Do you guys know this one? The statue, anybody in the room knows the statue? Oh, several, at least, where are you from? You Sabrina, you're German, right?
Yeah.
And you too?
Me too. Yeah.
So there are 2 in the room who
Yes.
so this is in the north of Germany, that's not important. There's a horse that is supposed to be in a swamp here. And there's a guy who's a baron. And he's actually pulling the horse out of the swamp, sitting on the horse. So the bootstrap idea is essentially you pull yourself out on your hair or on your boots, and that's a guy called Munchausen. From the Adventures of the Baron of Munchausen, there's an opera about that. And this is just one of those guys who had remarkable things happen to him, and that's the way to realize remarkable feats. And that's just one of those feats. And the idea of bootstrapping is called really after this idea. And it is simply the idea that you use the data that you have to do something that looks almost magical. So you take something out of the data that allows you to come up with an error estimate. And the way you do that, again, is you take your data set of, for this particular classification here, proteins between Golgi, extracellular, so the finally secreted proteins, and you have some split of your performance in your n-fold cross-validation. So you set on 5-fold all the proteins that you have, and for every single protein you know you got it right or you got it wrong. You have a set. And now what you do, say this set contained 500 proteins. Now what you do is you randomly pick 250 out. And you compile for those randomly 250. That's one way of doing it. And I argue is better than the traditional way of doing it. So for those 250 you compile how many of those do you get right. You do the same thing again. And you do it again. And you do it again and get an error estimate. That typically is much higher than the cross-validation experiment. And most likely much more realistic. Okay, now there are other methods out there. We discussed those, so we don't need to get into that one again. Now in these methods that also are based on machine learning, what we do here is now we want to compare our method to existing methods. How can you do that? Well, you could, in fact, of course, use the same cross-validation split, the same data set. That is not that easy, because not all methods are available. So, in fact, if you restricted yourself only to the methods that are available, you could possibly make a point. And you could argue, well, this is what is available effectively for the world. No matter what is published, this is what they can use. And there's some logic to that process. But it could be that one of the methods you have to pay for, right? And you in fact cannot pay because you don't have funding or something like that. And that really is a method that you absolutely want to compare yourself to. Ideally, you make your method available for free. I want to show people that by getting your method for free, they get something better than getting the other method that costs money. But in order for that to work, you still have to get the other method. Now, very often the methods that you can run like that, it could be a server somewhere. And running your protein through the server, you could do that. You could use your entire test set and run it through the server. What's the problem of that one? Well, p-sort, I just put it up, we talked about it from Paul Horton and others, may somehow be the least problematic of these issues. Say you wanted to really test the performance of Wolf p-sort. Wolf p-sort, Paul published a performance estimate for some data set, and that is the number. And you could compare that number to your number. Why can you not do that? That's apples and oranges, right? Because you have no idea what the data set is that Paul used. I mean, you have an idea, but let's assume you could actually access that as an internet server. And you could run your entire data set. So then you would compare apples and apples. Your test set, that method. What's the problem with that? Well, that method may have used the same thing for training that you used. And in a non-cross-validated way, right? So that method, in fact, you give your numbers for cross-validation. So you could go out and say, okay, then I compare, they had the chance in the past to use that for training, so I used my training and performance. Don't go there. That really is a bad idea. Many reasons why that is the case. Okay, so that you could do that, and you could argue that as long as you are better than that, you're fine. Because they, in fact, is unfair to you. If you look better, when you treat yourself unfairly, then that's a good way of doing it. Now, assume you don't look better. And you do want to look better. or something. Or, in this particular case, we had all kinds of reasons why log3 was a winner. So we were the first who predicted 18 classes. That alone, in some sense, since we could make the point that it really worked for 18 different classes, that alone would have been enough. But we wanted more than that. So we wanted to, this experiment will look better, we did that. But we wanted more than that. What else could you do?
Create a new test set.
How would you do that?
Let's use the redundancy of, for example, of sequences.
Mm-hmm.
So that you have a unique set.
Mm-hmm.
And then it would be best that the sequences were new.
What does it mean new?
After the method was created.
Yeah, this is exactly the idea. Now, when you say new Swissprot, and I have to make this point here, I'm not entirely sure whether that is considered already. So one way of calling annotations new is you simply say they have been annotated after the other method was published. Okay? But you have to also make sure that they are not similar to another protein that had been for which the annotation had been published. So there is an additional issue that has to be done, and I'm not entirely sure whether that was considered on this slide. And the set of all the proteins for which you apply in itself should be sequenced non-redundant. So it should not be just one family that you test. Okay? Now, having looked at all of that, you immediately see it's like a bacteria here, for instance. This is the standard error. What does that show you? Well, that's the estimate of how well p-sort works in a 5-state system in this particular case. That's the estimate how well log3 looks. Let's look at this one here, because log3 is more similar to the competitor. No, that doesn't work because it's our own competitor. So here we have one where the number for log3 is 65, while p-sort is 62. And it comes with a plus minus 14. What does that show you? Let's begin with a different question. The standard error is 14%. There's a Q9 estimate here. And again, we had to define the systems. log3 does 18 states, but this doesn't. So we had to go to the same number of states that they have here. We did not retrain. We just ignored everything else that was one of the other 9 states that were not considered here. So the first question is that number significantly higher, 65, significantly higher than 62 in that table? Can we make the point that we have a significantly better method? Is it yes or no?
If you depends on the method, one could come up with this bootstrapping scheme again.
Yeah, but it's there.
So you have
the answer to that bootstrapping scheme is on the slide.
But it would generate you a distribution. Right?
Yes.
So and you can
yes, that's true. But so I'm in a much simpler universe where I have of that distribution only one value, and that value is there. Or the standard error is the part that is. You can immediately relate the standard error to the standard deviation. So essentially that is the number that is here. So?
One can argue that...
No, just tell me yes or no. Absolutely not, right? Beware of that. You can now we get into, after we say, no, we cannot make that point, we can begin to argue. Yeah, but better anyway, blah, 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 blah. Now we can do all kinds of arguments. But we have to first off clearly make this point. Although 65 appears to be higher, is as a number higher than 62, I cannot, given this error margin, distinguish these two methods. Now the error margin is so high because in fact there were not that many proteins that came in new. for which I had annotations. So actually the height of the error margin now tells me that this is a clean comparison. So they are really new with respect to anything else that existed, plus there is a sequence unique subset. Now of these we had very few, that's why the error estimate is so high. That's why we can in fact not make that distinction, nor for instance the distinction between our old and our new method. For these we could use the same cross-validation folds, and we know that this is clearly much better than this, but we cannot see it in that data set. Here, however, in this particular comparison here, that's an outdated pre-version of Wolf-P sort that works for bacteria, so Wolf-P sort is not updated for bacteria. And here we see, despite the high, at least in some significant value, we can say, by the 10 minus 1 at least, we are clearly better. Okay? So this is more than 21%. No, we are not. I made a mistake. We are not. Sorry. Thank you. Thank you for being awake. Yes?
I wanted to ask.
Yeah.
Okay. Does this plus minus 16 mean that the error is not, obviously it must be skewed because 86 plus 16 would be more than 100, right?
Yeah, that is because the method can be very accurate. It can be over 100% accurate. Yes, you're right. But in fact, in an error estimate, that isn't. So this is not the kind of thing that I'd have ever seen people do. Plus 14 minus 16 would in principle be the right point here. And again, it would be a point to make the point that you understand that you cannot be 102% accurate. Other than that, you're right.
Yeah, but
Why could that happen? Why doesn't it come out of your... Because essentially it's an estimate. And it's a pretty wild estimate. So you can find examples where this is much, much more dramatic even for some of our small classes. To the extent that at some point, I'm not sure whether we get into the slides to that point, but for some of the classes, the error estimates is so high that there's no point of putting a number. Actually, we have a publication that puts a number because the reviewers forced us. But we strictly were against putting any number there. Okay, now here we have another aspect of it, and that is the aspect of removing the 30 N-terminal residues. That is the signal peptide here. C-terminal residues, ER retention motif, Golgi retention motif, and a third randomly removed. And again, in this particular case, this is a different data set. But in this particular case, what you see is that you lose, so that's the method for the full length. You lose by throwing things away. You lose somehow most for taking a third away and for taking the N-terminals away. That is again because the system in fact zooms into that signal here. Surprisingly little. But you also see that overall you still perform on what you may call an acceptable level. So again, so this is the lowest thing we are up against, and the lowest thing we are up against is something like 50% 18-state model. That's still surprisingly good, or whatever, the 9-state model, I believe this one is here. But this is still surprisingly good. And you do see that other methods tend to be a little bit more sensitive to it, to these reductions. Now, conclusion, we have a method that works fairly well, much better than anything else. And now we can use that method to, in fact, I told you, one missing aspect here is to put side blast back into the equation. So far we have done that only as a hack. That's what we call log tree 3. Now we can in fact use that and look at the composition for entire genomes. Here 6 of them are shown.